This video will serve to review the concepts of the cell cycle. The cell cycle is an ordered set of events which culminates in the division of a cell into two independent daughter cells. The cell cycle can be divided into two distinct phases, mitosis and interphase. A cell will spend the majority of its life during interphase. Interphase can be split up into three distinct sections. The first stage is G1, or growth 1, the first intermediate gap stage in which the cell grows and prepares for DNA replication. The cell's metabolism is heightened at this stage. The second phase, S, or synthesis, is when DNA is replicated. G2 is the last stage of interphase, in which the cell finishes growing and prepares for cell division by duplicating organelles. A way to remember all the processes of interphase is to memorize the mnemonic doctor. D for DNA replication, O for organelle duplication, C for cell growth, T for transcription and translation, O for obtaining nutrients, and R for respiration. Next, we will move into the mitotic section of cell cycle, mitosis. The first stage of mitosis is prophase. During prophase, DNA supercoils and is transformed from chromatin, the loosely packed form of DNA, into chromosomes, a tightly wound package used during interphase. Centrosomes will move to the opposite poles of the cell and start to form microtubule fibers. The last step is the nuclear membrane breaking down and the nucleus dissolving. The second stage of mitosis is metaphase. During metaphase, microtubule fibers coming from the centrosomes will connect to the centromere of each chromosome. Microtubule depolymerization causes spindle fibers to contract, and this contraction causes the chromosomes to align along the center of the cell, sometimes called the equatorial plane. The third stage of mitosis is anaphase. During anaphase, the continued contraction of spindle fibers causes the sister chromatids to separate. When separated, they're considered an individual chromosome in their own right. The separated chromosomes then move to the opposite poles of the cell. The final stage of mitosis is telophase. Once the two chromosomes arrive at the poles, the spindle fibers will begin to dissolve. Chromosomes will decondense into its previous form of chromatin. Nuclear membrane will reform around the chromatin. A cleavage furrow will begin to form along the equatorial plane of the cell. In animal cells, cytokinesis happens by the cleavage furrow, which is actually a contractile ring of microfilaments pinching off and creating two independent daughter cells. In plant cells, carbohydrate-rich vesicles will clump together along the equatorial plane after anaphase. This will form the cell plate. The cell plate will extend outward and attach to the cell wall, creating two distinct independent daughter cells. The regulation of the cell cycle is controlled by cyclins, a family of regulatory proteins. Cyclins activate cyclin-dependent kinases, or CDKs, which controls the cell cycle through phosphorylation. A cyclin and a CDK will form a complex, and together bind to a target protein and modify it via phosphorylation. The target protein will trigger some specific event within the cell cycle, and after the event has occurred, the cyclin is degraded and the CDK is rendered inactive again. While cyclins control and regulate the cell cycle, there are many factors that can disrupt the delicate process. A possible outcome of malfunctioning cell cycles is cancer development. Cancer is the unregulated growth of cells and can be caused by mutagens or oncogenes. A mutagen is an agent that changes the genetic material of an organism. They can be physical, chemical, or biological in origin. A physical example would be X-rays, a chemical example would be reactive oxygen species, and a biological example would be viruses, all of which have the possibility to change the genetic makeup of an organism and thereby disrupting the cell cycle. Mutagens that lead to the formation of cancer are classified as carcinogens. Oncogenes are the actual genes that have the potential to cause cancer that can be affected by mutagens. Most cancers are caused by mutations to two basic classes of genes, proto-oncogenes and tumor suppressor genes. Proto-oncogenes stimulate the cell cycle, and when it is mutated, it can cause increased expression in cell division, which can cause cancer. The second class, tumor suppressor genes, have proteins coded to repress a cell cycle progression. When these genes are mutated or are absent, it can lead to the unregulated division of cells,